Hello everyone, I'm Qianwen Wang, currently a postdoc at Harvard Medical School. Today, I'm presenting some preliminary results about our study, explaining deep learning approaches in drug repurposing through interactive data visualization. In this study, we propose an interactive visualization method to help biomedical researchers better understand and evaluate drug repurposing made by deep learning models. To start with, I will introduce some background information about drug repurposing and its role in drug development. Developing a new drug to treat a disease is a known and expensive process with a high risk of failure. It currently takes 13 to 15 years and $2 to $3 million to get a new drug to the market, and the costs are going up. Considering the high development cost and the safety risk of developing new drugs, drug repurposing is an effective alternative method. Instead of developing a new drug from scratch, drug repurposing aims to identify new therapeutic uses of existing drugs. Because most repurposed drugs have already passed certain phase of development and clinical testing, they can significantly reduce the safety risk and the development cost. It's estimated that the repurposed drug can get to market in less than half the time and at 1/4 of the cost needed to develop a new drug from scratch. As a result, drug repurposing draws increasing attention from both academia and industry. One famous example of drug repurposing is minoxidil, which was first developed for treating arterial hypertension. Then, due to the occurrence of unwanted hair growth during the clinical trials, researchers found that it can be used to treat hair loss. During the next decades, considerable efforts are made to repurpose drugs, but the number of successful cases is relatively small. Drug repurposing is still an open question, mainly driven by accident discoveries. Recently, deep learning models have emerged as a promising approach for drug repurposing. The increasing biomedical public data sets enable deep learning models to identify promising therapeutic opportunities at unprecedented speed, scale, and accuracy. Since most of those data sets present the existing biomedical knowledge as knowledge graph, in this study, we focus on graph neural networks, a type of deep learning models that are widely used in processing graph data. A knowledge graph is a collection of entities and their relationships. Nodes in the knowledge graph indicate different entities such as drugs, disease, proteins, and pathways. Edges indicate the various relations between those entities. Therefore, knowledge graph can effectively organize previously knowledge about drug repurposing. For example, previously, we have proposed a knowledge graph with diverse molecular data. This knowledge graph has in total 100,000 nodes, 7 million relations, covering more than 20,000 human diseases and 7,000 drugs. In this study, we use this knowledge graph as a training data set to predict new drug indications and provide visual explanations for those predictions. The GN model learns from the knowledge graph and predicts the relations between a drug and a disease that are not connected in the original knowledge graph. Therefore, the GN can identify unknown drug indications, contraindications, and off-label uses. However, as most deep learning models, the GN model used for drug repurposing acts as a black box. The lack of interoperability can severely hinder the application of deep learning to real-world drug repurposing. First, the GN model is built by machine learning specialists. The endpoint users cannot directly engage with the model, understand and assess the model predictions. Meanwhile, the lack of end-user engagement can lead to a biased or inaccurate model on certain diseases, wasting the efforts of downstream studies and even generate life-threatening consequences. Therefore, to successfully apply deep drug repurposing, it's crucial to provide explanations that can be easily interpreted by biomedical researchers in the context of drug development. There are several challenges we need to address in providing explainable deep drug repurposing. First, the provided explanations should be interpretable and actionable in the context of biomedicine. The explanation should not be a hunch that the users need to guess, but reflect clear biological mechanisms. Second, the provided explanation should be able to scale with different analysis granularities, including one disease and one drug, one disease and different drugs, a group of similar diseases, and the whole genome model. As mentioned before, the knowledge graph is usually huge and contains millions of relations. It's important that the explanations can summarize different levels of details. To address those challenges, we propose an interactive visualization tool to help domain experts understand deep drug repurposing. I will walk you through all the visualizations in detailed nature. Now, let's take a look at how a genome model predicts a drug indication for a selected disease. Generally speaking, a genome model tries to present each node in the knowledge graph as a high-dimensional vector, which is called node embedding. At each layer, the node embedding is updated by aggregating the information from its neighbors. After several layers, the GN is able to generate node embedding that reflects the properties and the interaction structures for each node. Those node embeddings are then used to predict the drug disease indications that are not known in the original knowledge graph. To help users better understand and evaluate the learned node representations, we present a node embedding view. For example, as shown here, all drugs are projected into a 2D space. We can see that those drugs with similar properties form clusters in the embedding view, indicating that the genome model is able to learn node embedding that accurately captures the node properties. Once a disease is selected, the genome model will predict drug indications that are not included in the training graph. The predicted drugs are highlighted in the embedding view. We can see that the genome model is able to predict diverse drugs for different selected diseases. To help users understand and evaluate the message passing, we propose a node attention view. The node color indicates the type of nodes. The edge cyclists indicates the importance of the neighbor information, and the tree structure reflects the hierarchical message passing. One node can have thousands of edges in the knowledge graph. Therefore, we implement a filter and only visualize important neighbors. Users can select different diseases and the drugs, modify the edge threshold. The visualization will be dynamically updated. While those two visualizations help users understand and examine the genome model, one problem is that those explanations are not consistent with the ones that the biomedical researchers would use to explain drug repurposing. Usually, biomedical researchers explain drug repurposing based on a series of biological interactions and functions. As a result, it's important to bridge this mismatch and provide explanations that are domain meaningful and reflect biomedical mechanisms. We propose explanation paths that are semantic paths in the knowledge graph. For example, ritonavir is a drug originally developed for treating HIV. Repurposing ritonavir for another disease, ALS, can be explained by this path, which indicates that ritonavir targets a protein that is implied in the disease ALS. We then use MatPaths to enable a consistent summarization of explanation paths at different granularities. A MatPath is a sequence of node relation types. It can summarize explanation paths for one disease and one drug, one disease and different drugs, or different disease and different drugs. The MatPath explanation also reflects how the GN model learns different relation types in the knowledge graph. As shown in the video, the MatPath explanation is able to present different explanation paths in an organized way. To better compare different predictions and their explanations, we propose a novel Mat matrix design. Here, each column is a predicted drug, and each row is a MatPath. The cell indicates the number of explanation paths for the predicted drug. Users can select one drug and examine the explanations in detail. Putting all those visualizations together, we design and develop a visualization tool to help domain experts understand deep drug repurposing. Users can search and select an interested disease, explore all the predicted drugs, Here, we demonstrate the proposed visualization tool using a case study where we predict and examine drug repurposing for Alzheimer's disease. The GN model used here was developed using the graph transformer architecture and trained using the previously mentioned knowledge graph. Then, this GN model makes predictions for drugs that are not included in the knowledge graph. To start with, users select Alzheimer's disease in the drop-down panel. The visualization is then updated to present the predictions and the explanations. The MatPath view contains different MatPaths, indicating that the GN model is able to consider a variety of biological mechanisms when predicting drug indications. The predicted drugs form several clusters and scattered in the node embedding view, indicating that the GN model is able to predict diverse drugs for Alzheimer's disease. Due to time limits, we will just investigate one drug cluster. We find all drugs in the highlighted cluster are anti-diabetic drugs, which is consistent with current scientific understanding of the relations between anti-diabetic drugs and Alzheimer's disease. In other words, the GN model is able to learn the knowledge about diseases and predict sense-making drug indications that are not included in the training dataset. We then select one drug in this cluster and explore the explanations in detail. While different MatPaths are provided, we close uninterested MatPaths and examine two types of MatPaths. For example, based on this highlighted MatPath, we can see that the drug Repurposing was predicted partially because it has the same target protein with some other drugs, whose target protein are associated with Alzheimer's disease. The two drugs that have the same target protein as Repurposing has also been reported to improve the development of Alzheimer's disease. Another important path is disease gene protein drug metapath, which indicates that the Repurposing targets as a protein that is associated with Alzheimer's disease. We found that similar explanation paths also exist for other anti-diabetic drugs. In conclusion, in this study, we design and develop a visualization tool to help biomedical researchers better understand and evaluate drug repurposing made by graph neural networks. We investigate the mismatch between model explanations and human explanations in the context of drug repurposing. We design a novel metapath-based visualization that provides domain-meaningful explanations and scales with the different analysis granularities. Thanks so much for your listening. You can find more details about the visualization tool at the online demo, and you are welcome to contact us to ask questions and let us know your feedback.